Welcome to the webinar. Today's topic is symbiosis, a multifaceted cocktail of signaling, touch, and sneak. Food. Who doesn't like food? Food is life. Food is core of culture. Food is precious. Food is energy. We have several definitions for the food. Recently, the importance of the food have been demonstrated clearly during the coronavirus pandemic lockdown period. I'm sure every one of us would have felt the heat of the food scarcity. However, the scientists have foreseen the food scarcity problem much earlier, probably 2 decades earlier. According to the population statistics, it's been estimated that by 2050, the world's population is going to touch closely 10 billions. One of the biggest problems of this century is feeding 10 billion people. How much more food we need by 2050? According to one of the recent reports, it's been estimated that we need approximately one-fold more, that is 100% more food production compared to the present food reserves. That means more or less this augmentation of the food production must be achieved only through the technologies. By adopting the latest technologies in the agriculture, this huge task can be achieved. This increase of the food production could be achieved more or less by 60% by targeting 4 important crops, that is maize, rice, wheat, and pulses. How to achieve this is the major questions. Nowadays, the farmers are excessively applying the chemical fertilizers in order to maximize the yield in most of the crops. Nevertheless, they are successful in getting the optimum quantities of the yield with applying the excess of these nitrogen fertilizers specifically. Check the fertilizer use imbalance map. This is one of the interesting study which shows that majority of the advanced nations such as United States, part of the Canada, Europe, India, and China uses the consumption of a maximum nitrogen per hectare is very high in these countries compared to the rest of the world. How good these nitrogen fertilizers are to the crop is an enigmatic issue. For example, let us check with the nitrogen transformation in the soil. For example, most of the agricultural lands, they apply the ammonium nitrates. For example, the ammonium nitrates are applied to the land, to the agricultural land. The crops usually takes the nitrates because of the high particle mobility compared to the ammonia, whereas the ammonia is slowed by plants. In meantime, the ammonia is further converted to the nitrates by denitrification method using some of the soil microorganisms. During that process, most of the nitrous oxides and the nitric oxides will be released into the air. It is one of the greenhouse gases. Secondly, the nitrates also will be converted into the nitrous oxides and nitrogen and nitric oxides by denitrification process. Therefore, the loss of the nitrates also occur within the soil and which releases further the greenhouse gases. One of the other method of loss of this ammonium also occur through the immobilization. Apart from that, the plants are capable to absorb the nitrates or ammonia largely depending on the season. For example, if the chemical fertilizers are applied during the summer, most of the ammonia is converted into the ammonia gas ammonium to ammonia gas through volatilization process. On the other hand, during the winter or rainy seasons, the solubility of the nitrates is increases, and therefore the availability of the nitrogen to the root rhizosphere will be decreased, because the nitrates will leached out, a process called as a leaching. It's leached down and it contaminates the groundwater level. So therefore, the application of excess of nitrogen fertilizers will definitely increase the yield. There is no doubt about it. But in addition to that, it is going to be so expensive to the environment. For example, it increases the pollution of the air and also contaminates the water. So what is the best alternative that the plant can use in place of nitrogen? So there are several methods and strategies have been applied. One of the thing which I personally feel that biological nitrogen fixation is the best alternative to supplement the nitrogen to the plants. Definitely here I don't rule out that this is the only method for alternative for the nitrogen, but this is one of the best method which I so the biological nitrogen fixation is not a natural process. It's not a new to the nature. It is always available in the nature, and we can see in the crop systems, and also we can see in the most of the cereals and in the legumes, in the tree species. Wherever you see the legume rhizobial symbiosis, this is the symbiotic association with the rhizobia, which is the major source for fixing the nitrogen, biological nitrogen. Some of the symbionts is not only present in the nodules, also you can find in some of the cereals. For example, you can see the associative bacteria, which is within the surrounding the root, also able to fix the nitrogen and augments the nitrogen capacity to the cereals. Similarly, the endophytic bacteria is another great source of fixing the nitrogen to the host plants. Let us understand little bit of nitrogen, biological nitrogen fixation in terms of taking one example. So here, this graph is a very interesting graph, where here the researchers have used two types of cultivars. One is the high yielding cultivars of the maize. The other one is in a cultivars which are very specific to the sub-Saharan African maize cultivars. Therefore, when the yield of the maize is optimum, the maximum yield will be achieved somewhere here between the white and the pink. Is the maximum yield is achieved in both the cases. How much nitrogen is required? This is the x-axis, shows that nitrogen fertilizer applied rate, that is kilograms per hectare. How many kilograms we needed to obtain the maximum yield is 134 kilograms per hectare. This is in case of maize. But whereas if you take any example of the legumes, it could be a bean, soya bean, or any of the medicago, and these medicago or any of the legumes have the capacity to fix the atmospheric nitrogen and also get more or less the maximum yield, if you can see here the graph, more or less the maximum yield without adding a single gram of nitrogen. That means it has the capability to fix its own nitrogen. How much it can fix? Approximately 120 kilograms per hectare nitrogen can be fixed on its own. So therefore, the scientists are largely focused to transfer this nitrogen fixing capacity to the cereals like maize. So that can solve completely the problem of pollution and also the nitrogen requirements for the plants. So therefore, the biggest challenge of this era for the researchers is to transfer the nitrogen fixing machinery from legumes to the cereals such as the rice, wheat and maize. This is one of the biggest challenges. A lot of funding in this direction has been done from past 10 years in order to make this as a reality. Let us understand a little bit about the mycorrhizal symbiosis. 80% of the land plants participate in this type of symbiosis. This is a fungal symbiosis basically. Most of the plants have the capacity to participate into the mycorrhizal symbiosis. However, there are some exceptions which are non-mycorrhizal plants. The well-known plants are Arabidopsis thaliana, a great model plant, and rapeseeds is an oil plant. Both Arabidopsis and rapeseeds belongs to the Brassicaceae member. Not only this, majority of the Brassicaceae members are known to be a non-mycorrhizal plants. Sugar beet is also another example of non-mycorrhizal plants. What is the benefits of mycorrhizal symbiosis to the host plants? The host plant gets benefited majorly in three areas. One is root system growth, nutrient efficiency, increasing of nutrient efficiency, water absorption. Root systems, basically the mycorrhiza secretes a unique chemical signal, something called as MIC factors, mycorrhizal factors. These factors have been proven very well with an evidences that these factors are responsible for the lateral root production. That means the lateral root branching increases when the MIC factors are added to the plants. So therefore, when the lateral root increases, obviously the rhizosphere for the plants increases and as well as the root biomass increases. And also when the early stages the mycorrhizal symbiosis occurs in the plants, then the establishment of the plant will be very faster compared to the non-mycorrhizal plants. Nutrient absorption efficiency increases dramatically in the mycorrhizal plants. For example, it uptakes the nitrogen, phosphorus, majorly the phosphorus, which is an inorganic phosphorus from the soil rhizosphere. It absorbs, and also the potassium and most of the micronutrients, which includes the barium, boron, sulfur and copper. Most of the essential micronutrients will be absorbed or uptaken by this fungi and transported to the host plants. In addition to that, the water absorption increases very high in the host plants, which gives an improved tolerance to the stress and greater resistance to the drought. So let us understand from this picture a little bit about the nutrient absorption and transport mechanism by AM fungi. Here you can see towards the left side you have a mycorrhizal independent pathway and mycorrhizal dependent pathway. So the roots, as always, the rhizosphere. The rhizosphere is in a peach color area here. The rhizosphere towards the left has a very limited rhizosphere. That means the area which is close to the roots. Whatever the nutrients, water available in this rhizosphere are directly accessible to the plants. Whereas on the right side, the plant is undergone to the mycorrhizal colonization. So in this, the rhizosphere is not limited only to the very close to the root system, but it is a widely spread. This is called as a mycorrhizospheres, because how much ever the area the mycorrhiza spreads, all that area is accessible to the plants to absorb the nutrients. Majorly what type of nutrients it absorbs? If you can see the color code here, inorganic nutrients and organic nutrients of nitrogen, phosphorus, potassium and water, and some of the micro nutrients as well. So therefore the mycorrhizal independent pathway has a limited resources compared to the mycorrhizal dependent pathway. So therefore the plants greatly benefits with the mycorrhizal dependent pathway, because the rhizosphere or mycorrhizosphere is much larger compared to the rhizosphere alone. So this is one of the example that how the absorption capacity of the plants increases when the plants participates in AM fungi symbiosis. Let us understand something about the rhizobial and mycorrhizal colonization, basically. So it happens in three stages. One is peak, friend and entry, or crosstalks, physical contact, invade. That's why it is, the title itself is called as, it is a cocktail of signaling. That means it is a crosstalks. The signaling happens at two levels in both the symbiosis. One is a chemical signaling and a molecular signaling. The second one is in a touch, which is nothing but a physical contact. They make some friends. Basically in this point, the rhizobium comes and contacts the roots, host roots, and mycorrhizal also germinates. The hyphae will contact the host roots at this stage. The final is a sneak, which is invade or entry into the system. That means once the rhizobia enters into the root, it penetrates into the inner or outer cortical cells, and later the cortical cells will divide and makes a de novo organ called as a nodule. This is where the nitrogen fixation occurs. Usually, if you can see, the nodules are in pink color because of the presence of leg hemoglobin. Only the functional nodules will have this leg hemoglobin. Therefore, they are the pink in color. In case of mycorrhization, once the fungus will come after the crosstalks or signaling is over, the mycorrhiza come and contact with the root surface, and later it enters into the root surface, and once it enters, that is no more called as an hyphae, it is called as an intraradical hyphae. So this intraradical hyphae ramifies within the outer cortex, and finally it will settles in the inner cortex close to the vasculature, and finally divides, something we can see, the finally divides like an arbuscule. So arbuscule is a finely divided, finely branched mycelia. So this is the place where the nutrient exchange occurs in the mycorrhized roots. So therefore, overall the symbiosis, whether it could be a rhizobial or mycorrhizal symbiosis, it occurs in the three major stages. And if you look at these evolutionary aspects of this symbiotic nitrogen fixation, fixing rhizobium and also the mycorrhiza, which are more recent, which is more older. If you can see here, the evolutionary concept, it's in a million years ago. For example, all the legumes, they have a parallel evolution. If you can see the red lines here, that indicates it's a parallel evolution. All comes under urocytes. Whether all these legumes have evolved somewhere close to the 60 million years ago. That means it's more recent compared to the mycorrhizal symbiosis, which based on the carbon dating and fossil evidences, it has been established that the mycorrhizal symbiosis is most ancient symbiosis, which dates back to the 460 million years ago. So therefore, the mycorrhizal symbiosis is the basis for the evolution of the legumes. That means much later, almost 400 million years later, the legumes symbiosis might have evolved from the pre-existing mycorrhizal symbiosis, is one of the theory, the strong theory, and which of course has the further, the scientists have proven with more evidences, and shows that yes, indeed, the mycorrhizal symbiosis is the basis for the newly developed rhizobial symbiosis. Let us understand that with this image. For example, the land plants, most of the land plants, for example, I told 80% of the land plants participates in the mycorrhizal symbiosis. In order to achieve this symbiosis, the plants utilizes a set of genes, a cascade of genes called as a symbiotic genes. Much later in the evolution, that means it is 460 years, and it is 460 million years. And much later in the evolution, from the land plants, legume groups have evolved. These plants evolved more recently. What was the age is 60 million years ago. So these legumes utilize the same set of symbiotic genes in order to achieve the rhizobial symbiosis process as well. So therefore, the legumes have the capacity to mycorrhize and also to rhizobial symbiosis. It can participate in both types of symbiosis, but it uses the same set of symbiotic genes in order to achieve the both mycorrhizal as well as the rhizobial symbiosis. This set of cascade of genes therefore known as a common symbiotic pathway genes. The legumes, the plants are so evolutionary, so sophisticated in order to use the pre-existing mycorrhizal machinery in order to achieve entirely a new rhizobial symbiosis as well. This is one of the best example, the legumes and the plant efficiency in order to conserve the resources in terms of new genetic elements. Sorry, it's a lipo-keto-oligosaccharides, okay, LCOs, and these are the lipo-keto-oligosaccharides, which is known as a nod factors. These nod factors are perceived by the roots with a special receptors, which is called as a symbiotic specific receptors. On the other hand, when the plant wanted to participate in the arbuscular mycorrhizal symbiosis, the plant roots will exudate strigolactones. This strigolactone synthesis will be induced only when the plant has a deficit of phosphate. So therefore, under the phosphate deprivated conditions, the plant roots secretes the strigolactones, and these strigolactones are perceived by the mycorrhizal fungi and they synthesize a MIC factors. MIC is a mycorrhizal factors, which is also a structurally different LCOs, lipo-keto-oligosaccharides. And these MIC factors are further perceived by the root receptors, which is something called as a MIC factor receptors of the plants. So these receptors are completely a different set of receptors for the mycorrhiza and for rhizobium bacteria. So once the nodule signal is perceived within the root, it upregulates a series of genes, a cascade of genes. These genes are, as I told you, it is a common symbiotic genes. So these are the SYMARK, castor, Polox, NUP, this is a nucleoporins, and the CCAMK and the cyclops. Once the signal passes through all these common symbiotic pathway, the genes, depending on what type of the chemical signal is perceived by the plant. For example, if it is perceived by the strigolactones, the CCAMK will activate specifically the transcription factor related to the mycorrhiza. If the signal is perceived by the nod factors, then the CCAMK, the same CCAMK, will activate the transcription factors related to the nodulation. So how does the CCAMK knows that? Which one need to be activated? Which transcription factor need to be activated? Because the CCAMK decodes a specific calcium spiking signals, which we are going to talk in the next slide. So once these transcription factors are very specific for the nodulation and specific for the mycorrhizal, once they are activated and they induce the genes as specific for the symbiosis of the mycorrhization and specific for the nodulation genes. So therefore, the plant will undergo changes to accommodate the mycorrhiza within the inner cortical cells, or the plant will synthesize a de novo organ called as a nodule. So this is the signal mechanism which happens in two parts: chemical dialogue and a molecular dialogue. Molecular dialogue is the downstream. So here the same the concept let us understand at a cellular level. For example, this is the rhizobium, this is the nod factors, these are the nod factor receptors. It is in a membrane bound, plasma membrane bound non-factor receptors with an extracellular domain. It binds here and it activates the SYMRK. This is a SYMRK. It is also a membrane receptor. On other hand, the mycorrhizal fungi, it secretes some MIC factors which will be perceived by, hypothetically, we are not sure about, MFR1, that is a mycorrhizal MIC factors receptor kinases, and this, once it perceives, this also activates the SYMRK. So both will activate the same SYMRK gene, which is a common symbiotic pathway gene. So once the SYMRK is activated, it transfers the signal to activate the NUPS, nucleoporin 85 and nucleoporin 113, and later it activates the castor and polax. These are the ion channel proteins which pumps the potassium ion in and outside of the nucleus. Therefore, as a result, the calcium oscillates in and out of the nucleus and cytoplasm. Therefore, the calcium produces as unique spikes. This is called as a calcium spiking. So these calcium spiking will be recognized by the CCAMK. This is the calcium calmodulin kinases. This is a very specific kinases which is present in the nucleus, which will be perceived, which decodes the signal. This signal, let me tell you, this signal is completely different for the rhizobium, completely different for the mycorrhizal. So therefore, the CCAMCO exactly know how to decode this, and depending on that, it phosphorylates with the cyclops gene and then activates the mycorrhizal transcription factors or the NOD factor or the nodulation transcription factor specifically. So this is the entire common symbiotic pathway. One more important thing I would like to add here is, once the rhizobium signaling is active, the defense pathway will be suppressed. So otherwise, the plant has a sophisticated mechanism in order to recognize whether this bacteria is a friend or is an enemy. So they only the friends, because it is a symbiont, it considered as a friend. Therefore, it switch offs the defense pathway temporarily, very short time. In the same way, the fungus, the mycorrhizal fungus, arbuscular mycorrhizal fungus, when interacts with this common symbiotic pathways get activated, and defense pathway will be shut down temporarily. So this is very important in order to recognize which is friend and which is an enemy. For example, the pathogens, the bacterial pathogens enters in the same mechanism, but of course not by the same receptors. In the same way, we have several fungal pathogens which also enters into the plant. In that cases, the defense system will be highly active in order to defend them. Okay. In this case, the symbiosis case, the common symbiotic pathway, when it is activated, defense pathway will be shut down temporarily. So this is what exactly I was talking. Calcium spiking is this is a best image which I can show you. This is a root hair where you have in the center the nucleus, and you have a red color spot which is the calcium signaling. So there are special confocal microscopes which can estimate or calculate the intensity of the calcium produced in both the rhizobial and as well as mycorrhizal symbiosis. And downstream to the mycorrhizal symbiosis, when the signal common symbiotic pathway is activated, downstream to that there are several genes. This is just I wanted to show you an example of genetic formation and development. I just wanted to give you a glimpse that how intense how many genes are part, more or less 100 genes participates. So far we know the 100 genes, and still are coming every week. The new genes are adding to the list. So there are several genes are grouped majorly into the depending on the type of the function. For example, bacterial recognition. These are the genes which recognizes the bacterial NOD factors or MIC factors. Common symbiotic pathway genes are early signaling genes. There are the list of the genes which are involved in the signaling or in the early symbiotic events. Later, the early transcription factors the genes which are related to the nodulation, and interactors which are essential for the induction of the early transcription factors and as well as the downstream genes. For example, the infection process. Infection is the entry of the rhizobium into the root hair cell. And nodule organogenesis. Once the rhizobium enter into the nodule, into the root hair, then it will go into the cortical cells and divide within the cortical cells, and then it produces a de novo organ, which is a nodule. So that is a nodule organogenesis. It is a synthesis of the generation of the new organ. So these are the sets, especially the cytokinins and auxins are involved in this process. For example, the nodule differentiation. So the nodule, within the nodule, the bacteria will be differentiated into the bacterioids, where they are capable of synthesizing the atmospheric nitrogen. They are the set of the genes which are involved here for nodule differentiation. Finally, the nodule number regulation genes. That means each plant, each legume has a specific number of nodules. How does it will be synthesized? How will it be regulated? It depends on the number of leaves and photosynthetic capacity of the plants. For example, if the plant is too big, which has a larger area of the photosynthetic capacity, that means lot of carbon is producing, then the plant will go for the more number of nodules. If the plant is very small, which a less number of leaves, then the carbon will be less produced through the photosynthesis. Therefore, the root produces less number of the nodules. That means that nodule regulation also involves lots of genes. So through this, you can have a glimpse that nodulation process involves several genes. So all these new functions need to be understood. In order, the basic idea, if you understand all these genes, then this knowledge can be extended to the cereals in order to engineer the cereals like a maize or a rice to be capable of synthesizing or fixing the atmospheric nitrogen. That is the ultimate goal for this entire research. So in this slide, let us check the developmental process of the nodulation. How the entire origin and death of the nodule, okay, from the birth and death of the nodule. Let us check it. So in the first diagram, which shows that there is a root hair. So once the common symbiotic pathway is active, then the rhizobium, the second stage is that when the rhizobium will come close to the growing root hairs. It's important, it's a growing root hairs, and get entrapped in the center of the root hairs, and the root hair get curls. So this is the curling. You can see the tip is curling. So at the center of the tip, the bacteria get entrapped, which is called as an infection pocket, where you can see a real-time image here. The infection thread is from the microscope. You can see the darkly stained body at the center, which is an infection pocket. It's a rhizobia. The blue color is a rhizobia because it's in a transformed rhizobia with a Gus bacteria, the gene. So therefore, Gus staining. So therefore, you can see the blue color here. So once the infection pocket is formed, the cell wall degrades. The host plant will secretes the cell wall lysis enzymes and degrades the cell wall, and the cytoplasm and the plasma membrane get invaginated. It bends into a hollow tube and grows inside like a tube. So if you can see here, the tube like growth you can see here. So there is still a plasma membrane surrounded by the bacteria. So this is a very important feature of the symbiosis. This is very important feature of the symbiosis, because though the plant gives access to the bacteria to enter into the plant cell, but still it keeps it away. Even though it is inside, it keeps it away because it is surrounded by the plasma membrane. So that means the plasma membrane, it is surrounded by whenever the plant wanted to give the food through the carbon, it has a control to give it. If you wanted to kill it at this stage, the plant will completely stop giving the carbohydrates through the plasma membrane. Therefore, the bacteria within that, it will die. So there is differently another process which is called as a nodule abortion. But this is the beauty of the symbiosis is, though the microorganisms enters into the cell, but completely it is under the control of the plant. So whenever the plant wanted to give the nutrients, it can give, and take the nutrients from that it can take. But the rhizobia do not have an access for any of the cellular machinery within the host root system. This is very important for the symbiosis to occur. So if you can see here, at 3 days post inoculation, that means once you put the rhizobium to the artificial conditions I'm talking about, once the signaling part, the common symbiotic signaling is get activated, the infection thread start forming at 3 days post inoculation. This is called as an infection thread. So once it enters, always it enters through the root hairs, never from the intracellular spaces in case of legumes. So once it goes into the epidermis and from the outer cortex cells, where you can see in the image C infection threads, and it branches out the infection threads, and also simultaneously you can see CCDs, which is cortical cell divisions. You can see here the cortical cell division, the cells are getting divided here. As a result, in the image D and also in the nodule primordia and the 7 days post inoculation, you can see a clear the division of the cells is occurring within the root. So this stage you can observe in the wild type roots at 7 days post inoculation. So once the nodule primordia is formed, the bacteria also start multiplying inside. So you can see in the image D, the blue zone is increasing. So first it was like a line, and now it is increasing intensely within the cortical cell divisions. Therefore, in 14 days, in this bit, the next 14 days, the young nodules will be formed, where you have a completely projected new organ, which is called as a nodule, and inside the rhizobium completely divides and occupies the cells of the cortex. And the stage, the mature nodules at 21 to 28 days, that starting from the 14 days to 21 days, the nodules will fix actively the nitrogen. So this is the stage where actually the plant get benefits in terms of nitrogen. Until 14 days, the bacteria is not able to produce any, not able to fix the nitrogen. After 14 days until 21 days, sometimes at 25 days, it keep on fixing the nitrogen within the nodule. And in return, the bacteria will receive malate, a large amount of the carbon source for the multiplication and growth of the bacteria. After 25th day or 28th day, if you can technically, the nodules go into the senescence. So once the senescence, the nodules turn to senescence, the color of the nodule changes from pink to green. That means there is no more legumoglobin. These nodules will slowly senesce and they die. Then the plant will initiate for the new nodules altogether freshly, depending on the requirement of the nitrogen. This is the entire process of nodule. So if you check the anatomy of the nodule at 21 days, we are taking here. Here I am showing the two examples. One is a determinate nodule and indeterminate nodule. So the difference is there are two types of the nodules. One is determinate, which is a spherical in shape, which has a definite growth, and indeterminate, which is like a cylindrical in shape, which at the tip it has a meristematic activity at the tip. So therefore the nodule will be keep on growing like a finger like structure, very tiny. So therefore it is called as the indeterminate nodules. So in both the cases, the nodule anatomy remains the same. For example, I wanted to show you here the figure E, where within the nodule transverse section, if you can see, the darkly stained, the blue cells are the infected cells, and compared to there are the white small cells, which is called as uninfected cells. So the bacteria is technically present in the infected cells. So this one of these infected cells, we did it electron microscopic studies, which shows that this is the infected cells. Within the infected cells, there is a peribacterial membrane. So if you can see, a membrane is there within this peribacterial membrane, which is called as a symbiosome. So in that you have a bacterioids. So symbiosome is as in which you have five or four. It depends on numbers, and you have a small bacteria. So these are called as a bacterioids. So this is the site where the bacteria are capable to fix the atmospheric nitrogen. Once it fixes, it will give back into the cytoplasm of the infected cells, and it will be transported back to the vasculature and phloem or xylem, and transported to the shoot part. Okay, so let us understand the mechanism of the nitrogen fixation a little bit. Okay, briefly I wanted to touch this aspect. You have a xylem and phloem, and through which the sucrose will be pumped into the infected cells. For example, this is a symbiotic cell. The large cell is a symbiotic cell. Within the symbiotic cell, you have a small cell which is called as a symbiosome. So this is what I was telling. The symbiosome, this is a membrane of the plant. So therefore, whatever the nutrients the plant wanted to give it to the bacteria, it has to pass through this membrane. It is regulated by the plant completely, the host plant completely. Within the symbiosome, you can see another small structure, which is, this is called as a bacterioid. So this is a bacterioid. Bacterioid has the all cellular components. For example, when the sucrose is pumped into the infected cells, specifically a malate, which is a 4-carbon malate, will be pumped into the will be transported into the bacterioid through the symbiosome perisymbiotic membranes, and then this malate will enter into the tricarboxylic acid cycle, and then produces enormous amount of ATP. So this ATP is very important. Why? The nitrogen fixation is a very expensive process, which requires, for example, if we see in the beginning, to fix the one molecule of the nitrogen, it needs a 16 ATP molecules in order to get 2 ammonia. So two ammonia molecules, for example, basically it produces ammonium. So therefore it needs 16 molecules of the ATP. And what else you need is, you also need a nitrogenase is an enzyme, is a very important enzyme present in the bacteria. So it takes the nitrogen gas, and in presence of nitrogenase and the high amount of the energy, it synthesizes the 2 molecules of the ammonia, and it will be transported back into the host cell. This is the infected cell cytoplasm of the host cell, and then it will go back to the xylem and phloem, and then to the wherever the nitrogen is required in the shoot or root, it will be transported back to there. So this is the overall mechanism of the nitrogen fixation within the nodule. So this nitrogen fixation capacity will be very limited. Like, it starts from 14 days and it will be end at the 28th day. So therefore, within that period, the bacteria will keep on. So once it is over, then the bacteria will go for the nodule will go for the senescence. So let us understand a little bit of the legume nodule types. Okay, this is just to give an idea that how many types we have. We have a determinate type of nodules, desmodioid types, which is a spherical. Nodules are very spherical. For example, the common bean Phaseolus vulgaris is you can see the spherical nodule. It's a determinate type of nodules. Another one is determinate ascinominoid types, which also has a spherical shape, and which belongs to the different families of the plant families. The other one is indeterminate unbranched. So it is an indeterminate, but it doesn't have any branches. It is specific to the some group of the plants. And indeterminate, one or few branches. If you can see, it is like a 1 or 2 branches you have. That means it has 2 meristematic zones. Here the only 1 meristematic zone. So this is very specific to the papillonoid groups. And indeterminate desmonoid types also you can see. This is also something like an unbranched indeterminate type. And lupinoid nodules. Lupin is a plant, a legume plant, which is specifically, it has an amorphic type of lupin nodules. Then indeterminate with many branches. and where you can see it is like a peg-like structures, and it's like mushrooms growing on the root. So it's indeterminate with many branches. Finally, you also have woody nodules in some of the tree species, which also an indeterminate type of nodules. Apart from that, a few legumes such as a Sesbania rostrata have a nodules, very unique nodule. This plant has two types of the nodules. One is on the stem, another one are normal nodules on the root. In the stem, as a unique nodule because these nodules are green in color. It doesn't mean that they are senesced, but they are green in color because it has a capacity to it photosynthesize and also fix the nitrogen. That means it is a very unique nodules you can find in this legume. Apart from that, as a recent interesting study, this is a very important for engineering nitrogen fixing cereals. Nitrogen fixation. These are the very unique land races of Mexico. In Mexico, these land races of maize was found, and very recently they have published this paper, and they did almost a decade of work in order to understand the whole concept of nitrogen fixation here. And this maize can fix the nitrogen. How it fixes? It has advantageous roots. So these advantageous roots uniquely secretes, only in this land it secretes lots of mucilaginous, where you can see lots of mucilage around the advantageous roots. This mucilage is rich of different types of carbohydrates like xylulose, fructose, arabinose, mannose, galactose, and all this. So also have some of the unique microbiota, including some of the rare species of the rhizobium, and which can fix the atmospheric nitrogen directly, and with some unknown mechanism the plants will absorb this nitrogen. So this is one of the best clue that yes, it is possible to extend the nitrogen fixation capacity from the legumes to the cereals. So that is one of the good news for the researchers who works on this. So now let us talk about something about the mycorrhizal symbiosis altogether. So developmental process of the mycorrhiza, let us understand a little bit of the mycorrhiza. And this one thing I would like to is a very unique nature of the mycorrhizal spores is the spores is a capacity to germinate 10 times in its lifetime. That means once the spore germinates, it will go in search of the root. So once if by accidental by any chance the hyphae doesn't find the root, then it will withdraw all the cellular content, and then it will germinate in another direction completely in a new way. So that means each spore has a capacity to germinate and withdraw for 10 times. So that means it is a very resistant spores. And if you can see this diagram where you have a spore, after perceiving the strigolactones, and then it produces the MIC factors, that means the chemical dialogue is over and also the molecular dialogue is over. Now that is a point of physical contact. The spore will germinate and come in contact with the root surface. So once in contact, and it produces a seed and a seed-like structure called as a hypopodia. And this hypopodia, which gives a good anchoring for the mycorrhiza to attach on the root surface firmly. So just below this hypopodia, there is a unique structures called as a pre-penetration apparatus, which will be which you can see here with an arrows, which in the striped regions in this cell, which is something called as invaginated plasma membrane, which is called as a pre-penetration apparatus. So it invaginates, it gives a halo kind of structure, and which gives a passage for mycorrhiza to enter. So this will be predetermined. So before the mycorrhiza enters, this cell will be preparing this kind of PPS. So this pre-penetration apparatus is like a guide for the root in which the fungi is supposed to travel. Okay, so here once it contacts the hypopodia, will then it germinates, and this tip, usually the mycelia grows at the tip. So in the tip will go on ramify in the extraradical hyphae, in the intraradical hyphae within the outer cortex of the roots. So at the end, when the intraradical hyphae reaches to the inner cortical cells, it finally branches and produces something called as arbuscule, where you can see here in this in the confocal image, the green structure is an arbuscule, whereas it initiates from here. This is in a hypopodia, and then this is an extraradical hyphae, where you can see a dark structure is a extraradical hyphae. Once it enters, it becomes an intraradical hyphae. It ramifies in the inter and intercellular spaces within the cortex, outer cortex, and once it reaches to the inner cortex, where you can see it is very close to the vasculature, these arbuscules will be formed. So this is how the mycorrhiza enters and colonizes the root of the host plants. So if you can see the AM fungal development in the root, like in a timeline period, for example, in the first week of the infection or colonization, you can see here, this is the entire thing called as a one infection unit. So the extraradical hyphae comes and makes an hypopodium, and then enters intraradical hyphae and produces the arbuscules. Next, and within 3 weeks, you can see lots of the arbuscule numbers increases throughout the root, and it intensifies in 6 weeks. In the close-up image, you can see the intensifies. Almost all the cells will have the arbuscules, and this is the structure of the arbuscules, and a close-up structure which was stained by the Alexa fluor, and it's in a different technique to stain the roots with using the fluorescent Alexa fluor. And also the mycorrhiza can be spored with a trypan blue. Okay, in this case, these 4 images with the trypan blue, and this is with an Alexa fluor. So this is how the mycorrhiza, and it doesn't have the senescence actually, but if the arbuscule function is over, this arbuscule will collapse and just degrade within the cell. But however, the entire mycelia will not be disturbed. It will form the same mycelia, intraradical mycelia, extend to the another part of the root and forms a new arbuscule. But the collapsed arbuscules will be absorbed back by the plants. So if you understand the mechanism of the nutrient uptake, this is a very simple mechanism. You have a plant here, the plant cells, and you have a membrane, plant membrane, and you have a fungus within the plants. For example, this is, I'm talking in the arbuscule. Within the arbuscule, this is a plant membrane. In the plant membrane, you have a variety of the transporters. For example, the sucrose transporters. Sucrose is the main source the plant gives to the fungus for its growth and development. In return, the plant will uptake the phosphorus and nitrogen and some of the other micronutrients as well. And it has several in the plant membrane. If you can see, the plant membrane has several transporters of the ammonium nitrate, amino acid transporters, urea transporters, and also the phosphate transporters. So the inorganic phosphates, whether it could be polyphosphates or the pyrophosphates or orthophosphates, will be uptaken by these receptors, and then it will transport them back to the arbuscule. Within the arbuscule, it will give to the plant. In exchange, the fungus will receive the surplus amount of the carbon to the fungus. According to a study, it is been shown that 20% of the photosynthetic carbon will be given to the mycorrhizal fungi. So that means it's a huge commitment for the plant once if it enters into the AM symbiosis. So if you talk about, if you look at the types of the mycorrhizal fungi, you have 5 types basically: ectomycorrhiza, ectandomycorrhizas, ericoid mycorrhizas, orchid mycorrhizas and arbuscular mycorrhizas. So what is the major difference? I wanted to just you must be knowing this aspect. This is the number of species, like most of the tree species and in the forests, and some of the shrubs and trees will participate ectondo and ectomycorrhiza. Both will have the extra radical hyphae and a thick mantle. A mantle is a thick hyphal structure. And what is the difference between the ecto and ectando is with the ectandomycorrhiza, you also have the pilotons, which is in a highly coiled structure within the outer cortex. Whereas in case of ericoid, where the most of the berries are undergoes this kind of mycorrhizal colonization, which also shows the extra radical hyphae and the pilotons. And in orchids also, it has the same structure like the ericoid mycorrhiza. Whereas what is the unique thing is the arbuscular mycorrhiza, where most of the crops and the shrubs will herbs will participate this kind of arbuscular mycorrhizal symbiosis, where you have an extra radical hyphae, intraradical hyphae, and also vesicles and orbicles. So this is the only unique mycorrhizal, and large groups of the plants participate this type of mycorrhizal symbiosis. This is one of the interesting slide which I personally like. The mycorrhizal network. How big the mycorrhizal network is an interesting question. So far we know that which is the biggest organism in the whole world is the blue whales. The blue whales is a very huge, and they are the largest living organisms. But now it's not true. Let us see that. The largest known mycorrhizal network was found recently in the Blue Mountains of the Oregon in the United States. And this covers something like 3 square miles. The single mycorrhiza spore germinates and infects all the tree species and the shrubs and the herbs present in that 3 square miles area. Only one spore. And all is like one single giant organism. So therefore, now no more a blue whale. The mycorrhiza itself is the biggest living organism on the earth. So it has been estimated that if you can carefully take out all this mycorrhiza mycelium and collect them and weigh them, it could weigh something like 7,500 to 35,000 tons of weight. It's very lot. It's a huge lot, because the blue whale it's only counts for 115 tons. So there is no doubt that the biggest and largest living organism on the earth is the mycorrhiza. This is one of the interesting finding. And now finally, the mycorrhizal and arbuscular rhizobial and mycorrhizal symbiosis are like 2 eyes of the agriculture, because the nitrogen and phosphorus are very, very essential nutrients for the growth and production of the crop, the production of the croplands. So therefore, to lay the path to the nitrogen fixation cereals. So this is the ultimate goal. The ultimate goal is to transfer the nitrogen fixing machinery to the cereals. So to lay the path to the nitrogen fixation cereals, first we need to understand the function of the symbiosis related genes in legumes. So first we need to understand in legumes and understand all the functions related to the symbiosis and in both the symbiosis, and then you extend that knowledge to the cereals. So let me give an example. We have done a lot of work in this direction. I just wanted to present only one paper which we published in the Plant Cell, and this is a gene called as the PI3K. So this gene is an autophagy related gene. When we silence this gene by RNAi technology, where you can see this ABC is the control, and D, E, and F are the silenced plants of the PI3K. So in this, once the infection thread, the rhizobial infection thread is formed, it branches in the control like normally, and also have the cortical cell divisions. Whereas in the lower panel, you see the infection thread very well, it is branched until here, and beyond the epidermis it is not passing through. There is no cortical cell divisions we observe. That means it is hampering in the early stages of the nodulation process. Similarly, we also studied the same gene under silenced conditions in the mycorrhizal symbiosis. So where you can see, so this is the control plant, the first A. So where you have the hypopodia and then the intraradical hyphae, and it have a lot of arbuscules formation. Whereas when the PI3K is silenced, what is happening here, if you can see in the image C, PI3K, when it is silenced in that root, the mycorrhiza enters into the root by normal way, by hypopodia, and the intraradical hyphae, as soon as it enters, it forms a bulged tips. The tips of the fungi will get bulged, a finger-like structures will form, and that is the sign of the abortion. So once the entry takes place, immediately it gets aborted. So that means it is not going for the AM symbiosis process when the PI3K is silenced in the roots of the common bean. These studies we did in the common bean. So that shows that our model, this PI3K gene acts downstreams to the common symbiotic pathway. If you can see this common symbiotic pathway, remember the common symbiotic pathway which is common for both rhizobia and also for the AM fungi. Then downstream to that, when the PI3K is silenced here, when we silenced here, we do not form the arbuscule maturation. Why? When PI3K is silenced, it doesn't upregulate the transcription factors like a RAM1 and RAM2. Therefore, no mycorrhization is happening. On the other hand, downstream to the common symbiotic pathway, when the PI3K is silenced during the nodulation, infection progress is aborting and also the organogenesis is affecting. Therefore, nodulation is affecting. So that means the PI3K is it acts like a common symbiotic downstream to the common symbiotic pathway, which is very essential for both nodulation and also for the mycorrhization in common bean. This is the conclusion we get. So this is the new function we identified. Now this knowledge will be extracted, and we will be extending to the cereals and check how this when you overexpress these genes, how the cereals are going to behave. Will it go for the nodulation? So that's what our basic idea. So since because of that time concerns, so I just wanted to give you the list of the genes. So far we have done we have identified the novel genes related to the symbiosis process, both mycorrhizal and as well as the nodulation. So these genes have a different roles in these two symbiosis. For example, RBOHA and RBOHB, these are the ROS generating genes, which is very important and essential for both types of symbiosis. Annexins, NIN is a nodule inception genes, which is important it's an important transcription factor, which is without this the cells will not go for the cortical cell division will not occur. And later tetraspanins, and also the SNRKs, a sugar regulating genes, and TAR, triargutatapamycin, is a master gene, and this also has a role in both symbiosis. And flotilin, CRKs, and BPS, and several genes we are working, and we also already got a very interesting results, and which we are also working using these genes and extending this knowledge to the cereals, and to check whether how these genes are acting, and is there any chance that the cereals can invade these rhizobium bacteria within the roots and fix the nitrogen. So far we have got an interesting observation in sorghum as one of the cereal. So hopefully we are going to do much more in this direction. Okay. Thank you very much for your patience and attention, for listening all this. If you have any questions particularly related to this talk, please do write to me in this email which I have given here, and also if you have some time, please check this website where you can see my work and my publications and groups, and if you are interested in this area. Okay, thank you very much.